Ma'am, you can start.
Sir is not audible. Bhupesh sir.
He is not connected to the audio, ma'am. We need to connect it.
But I told him.
Okay, ma'am.
You have his number, no? Pradhamesh, can you call him? WhatsApp call. Good morning, audience. Bhupesh sir, good morning. Hello, Bhupesh sir. Pradhamesh.
Hello.
Hello, good morning,
Hello.
sir. Bhupesh sir.
Good morning. Good morning. Yes, ma'am. Am I audible
Yeah.
to you, ma'am?
Yes, sir.
Okay. Okay.
Shall I start, Pradhamesh?
Yes, ma'am. Yes, yes.
Yes, ma'am.
Good morning, Bhupesh sir, and all the faculty members, research scholars, students. On behalf of Microbiology Society India, I, Dr. R. Ushasri, from Chennai. Welcome our distinguished chief guest and the speaker of today's awareness program, Dr. Bhupesh sir, who is assistant professor, Department of Forestry, School of Science, Nagaland University, Lumani, and to his credits, he was associate director, senior research scientist, King Institute of Preventive Medicine, and he was junior research fellow, lecturer, and head of the department for EGSP College of Arts and Science, Nagapattinam. And to his credits, he had obtained many awards like UGC Meritorious Scholar Award, Young Scientist Award, Best Paper Award, Noveler Award, and he was his professional. He is a member of many professional institutes and academic associations. He has attended many programs and in international conferences and national conferences. He published 118 publications, and it is our privilege to have you, sir, for this awareness program. Thank you so much, sir. Kindly start your session, sir. Thank you, sir.
Yes, ma'am. Yes, ma'am. Thank you for your nice introduction on me, ma'am. Am I audible to you, ma'am?
Yes, yes, sir. Yes, sir.
Am I audible to you? Yes. So first of all, this is my first presentation in Microbiology Society of India. So I am the life member of Microbiology Society, and I am very grateful and thankful to our President, Professor Deshmukh sir, Secretary and Vice President, and all other state presidents for giving me this opportunity to present my lecture on antimicrobial peptides and its delivery through nanoparticles from the catfish Clarius batracis. So I am Dr. Bhupesh Giridharan, so who is working in assistant professor in the Department of Forestry in Nagaland Central University, Dumami, Zunibito. So now the present slide shows that the focus of the infection surrounded by epithelial cells, so and how the defense mechanism is against the infection in the cell. Okay, so here the beta defensin, this is the beta defensin which was stained by brown color is the peptide. That is the peptide which is present in the non-specific defense system of the immune system of the fish, which is called Clarius batracis. See, in nature, we have plenty of defense particles, defense system. So those particles which always fight against the pathogen and control the pathogenic infection from the host. So that here some of the antimicrobial peptides. First of all, what is antimicrobial peptide? So antimicrobial peptides are the ancient weapons. This weapon is the natural weapon which is present in the prokaryotic system, eukaryotic system. So especially we focus on the peptides which is present in the animal and plant kingdom. There are plenty of antimicrobial peptides that have served a fundamental role in the successful evaluation of complex multicellular organisms. So these peptides are amphipathic peptides, which is a cluster of hydrophobic and cationic amino acids which are spatially arranged. Those peptides acts as a allelic function, more than multiple functions the peptide is doing. This peptide acts as the signaling molecule to perform the post-translational modification during the proteolytic processing and the glycosylation. And these peptides are acts as number one defense peptide in our innate immune system. So like many peptides are there, So the peptides in even in the curd also there is an important peptide which is called lactoferrin that is present in the milk also. So similarly, there are plenty of peptides are there. These peptides are small molecular weight proteins, so which have a broad spectrum antimicrobial activity against bacteria, viruses, fungi, and even transformed in the cancer cells to fight against the cancer also. That way these antimicrobial peptides has anti-cancer properties. So shortly, these antimicrobial peptides, we call it as in the pharmaceutical industry, we call as the peptide antibiotics. So in diabetes, we are taking insulin to control the diabetes. Similarly, these peptides are the number one peptides which have a specific role in controlling the microbial infections. So now we see about the classification of antimicrobial peptides. There are beta sheet, alpha helical, loop, and extended. Four different structures of peptides are there. So let me see one by one. See here, this is the alpha helical peptide, which is a specific class of peptide called meganin, which is present in the frog protein, frog immunity system. And this is the indolicitin, which is present in the insects. This is the protein, which is present in the insects. And this is the defensin, which is present in both human as well as frog. So these are all the different structures of antimicrobial peptides. So
Sorry to disturb
we
you, sir.
have
The PPT is
identified.
not visible.
Oh, 1 minute, ma'am. Hello, hello,
So it's getting started,
hello.
sir.
Okay.
Yes, sir, it is visible now.
Yes, ma'am. So there are different types of antimicrobial peptides which is already identified in different both the in eukaryotic plant system. So I have listed some of the antimicrobial peptides in these slides, like thionin, which is identified from the plants, which is both the broad spectrum antimicrobial property to control bacteria and fungi also. Plant defensin, which is identified from the plant, which is having the broad spectrum antimicrobial activity. Now it is also another peptide which is identified from the plant. Antimicrobial peptide, which is identified from the insect, which is having, and then insect defensin, prosomycin, clavanin, statins, cathepsin, and pinadins, and the lipotoxins. So these are all the variety of peptides which was identified from plants, insects, and animal and aquatic system also. Hello. So there are three human antimicrobial peptides. They are defensin, cathelicidin, histatins. So defensin is found in neutrophils, panic cells, monocytes,
Sir, again the PPT is not
macrophages.
visible.
Oh, ma'am, maybe some network issue, I think, ma'am. So 1 minute, I will connect. Hello, am I audible? Hello, hello.
Yes, sir. Yes, sir.
Yes, thank you. Some network issues here. Sorry, because today I am presenting from my home. If it is from the university, I think uninterrupted network is there. Okay, so sorry for that. So that this cathelicidin are found in secretory granules of neutrophils and in natural killers and T cells and mast cells and epithelial cells. Cystatin is another smaller antimicrobial protein that are full of histidine and are found in the human saliva. So the mechanism of antimicrobial peptide. So how it was targeting the how what it and so this is mechanism. There was two type of pathogenic
Unable to hear anything. Can somebody say whether they can hear?
No, sir, it is not audible.
Exactly, yeah.
I think it's Bhupesh sir's audio is not connected, sir.
Yeah, we can't hear anything.
Yes, sir, right.
Somebody can telephone and tell him otherwise. Very interesting topic, but the thing is no audio, so that becomes the problem.
Yes, sir, right. That's right. Pushastri ma'am is there, but I guess she's not there in the meeting.
Your name is what?
Sir, this is Rupa here, sir.
You are talking from where?
Sir, I have connected from Mumbai.
Okay, okay. No, because now this speaker is from Northeast, no? Nagaland, I believe.
Yes, sir, right.
Even in West Bengal also right now connectivity issue is there.
Yeah, that's true, sir.
The speaker is G. Bhupesh, right? Rupa?
Yes, sir. Speaker is G. Bhupesh sir only. Yes.
Yeah, but if you see, that mute, it is showing some different symbol also after his name. Some green
Sir,
color is
actually,
there.
sir's audio is not connected. Only video is connected and it is off.
Yeah,
And audio is not connected.
sir, actually I'm coordinating, but he's not responding, sir, even in WhatsApp. What shall we do, sir?
Now you telephone him on mobile and let him start from the beginning.
Okay.
I think that is the best, no? Because if this presentation, if it is recorded, then it is not good. Only our conversation will be there. You can telephone to his mobile. Some 50 slides he wants to present. We are somewhere now in the 18 slides. Oh. Connection also has gone now.
Hello. Hello, Bhupesh sir. Bhupesh sir. Unmute your audio system, sir. Bhupesh sir. Hello. Hello. Who is Rupa? Allow me, sir.
Yes, ma'am. Yes,
Tell me.
No,
ma'am.
I called him, but he doesn't, I think, how to connect. What?
No, what he can do is he put up and then log in once again.
Yes.
That
Yeah,
is
that's
the,
what
Exit
I
करके
told
फिर से
him.
login करेंगे.
Yeah, I told him. Yeah, now just now
नहीं,
I updated him again.
उसको बोल दो कि लॉग ऑफ कर लो
Ma'am,
करके और फिर
audible to
रिज्वाइन
you?
करो करके.
Now he is
Hello?
bit audible.
Yeah, now, yeah. Bhupesh sir,
Yes.
can you hear? Yeah, please start, sir.
Yes, yes.
भूपेश थोड़ा सा शुरू से ही करो briefly.
Yes, sir, yes, sir.
Now you are in which place? Are you in Kohima or what?
I'm in Lumami, sir, headquarters of Nagaland University.
Kohima.
Sir, am I audible to you, sir? My slides are visible to you?
Yes.
Yeah. Now it is better, but then it shows still little bit problem. Maybe we will adjust. Maybe what you
Okay.
do is you can put up your audio, your video.
Yes,
Clarity
sir.
will be better. You put up your video and start only the presentation.
Okay, briefly restart,
Yes.
sir. Bhupesh sir.
Exactly.
Okay, thank you.
Oh.
By
Yes.
12-15 minutes, you can finish up, sir. You can wind up because you have Bhupesh sir has
Yeah.
another schedule by 12 o'clock. Thank you.
Yes. Ma'am, now my video and audio is reached you now?
Sir. Yes. Please start,
Okay.
sir.
Yeah. Sorry, actually, I'm presenting continuously. So if Rupasri ma'am didn't interrupt me, then I don't know whether my connection is on or not. So I'm going on in presentation. Okay, so this there are three important antimicrobial peptides we have already saw. That is defensin, meganin, and cathelicidin. The major function of these antimicrobial peptides are the key function is antimicrobial action. So that is killing the different broad spectrum microbial organisms such as bacterial, fungal, and viral. And it is also having the immunomodulatory effects. So, am I audible to you? Hello?
Yes.
So then cell apoptosis. So cell apoptosis is a programmed cell death. So it is also an important mechanism which is to target and kill the pathogens, and it is also having the antimicrobial activity. So my research experiences. So how I am specialized in the antimicrobial peptides. So in my entire research career, I worked on these antimicrobial peptides, which I have already isolated from the catfish Clarius batrachus. So I have discovered a new peptide which is called fluorocidin-like peptide, which was isolated from the Clarius batrachus fish. And another one peptide which I isolated was meganin from the Mirgal Sirhinus cirrhosis in my PhD studies. And defensin, a non-specific defensin, a non-specific immune peptide which was isolated from the frog Rana clamitans in Nagaland University. So these antimicrobial peptides are very specific in the antimicrobial property as well as anti-cancer property as well as wound healing property, and also it acts as the signaling property. So these peptides are delivered through a specific nanocarrier such as nanoparticle. So the functions and the delivery of the peptide is sustainable. So that I have selected an important particle which is called chitosan nanoparticle. So you know that chitosan is a safe, non-toxic, biodegradable polymer from the chitin. It is the second most important and abundant polymer. The first is, that is the starch. It is a positively charged, and you can make it as negatively charged surface. You can make the surface modification in the chitosan polymer. It's soluble in diverse acids to interact with the polycation to form complexes and gels. So these are the different sources of the chitin. Usually chitosan is a polysaccharide, where the polysaccharide is isolated from a important source which is called chitin. The precursor of the chitosan is chitin. If you deacetylize the chitin, then by acetylation of the chitin, which will leads to the synthesis of chitosan or a product called chitosan. You can able to extract the chitosan from the prawn, crab, lobster, crayfish, shrimp, squid, and mushroom. So this is the structure of chitosan. How the chitosan is synthesized from the chitin is by deacetylation in sodium hydroxide 45% to 80, 45% at 80 degrees Celsius. You can get the chitosan molecule. So the
Dr.
chitosan
Bhupesh,
nanoparticle.
Dr. Bhupesh,
Hello.
we are unable to see,
Yes,
we
sir.
are unable to see your PPT. Maybe your audio you can put it up, video, and presentation you can share.
Yes, sir. 1 minute.
Otherwise, very useful lecture, very nice presentation also.
Yes, sir.
We can see you actually right now.
1 minute.
We want to see your PPTs also.
Yes, sir. Yes, sir. Sure, sir. Sure. Sorry, actually, now I am presenting from my
You are from
home. So
you are from Tamil Nadu or Andhra?
Tamil Nadu.
Which place?
Sir, now, sir, is it visible, sir?
Not yet. Yes, wonderful, wonderful. Please go ahead. 27th
Oh,
slide
yes.
number we are in.
Yes, sir. Yes. Yes. So this is soluble in acidic solutions in the free amino groups. So it's giving positive charge. We can modify the surface of the chitosan nanoparticle. So by different ways. So we have in my research, I have synthesized 4 types of chitosan, such as tripolyphosphate chitosan, carboxymethyl chitosan, N-succinyl chitosan. So this chitosan molecule, which is biocompatible in nature, it is less toxic, and then it is non-immunogenic, and then it is biodegradable, biocompatible. It is also having, if you modify the chitosan nanoparticle, it is also having antimicrobial property, and it absorbs the toxic, and it is having the good binding property, and it is having immunostimulating activity also. So these are all the different means of synthesis of chitosan nanoparticle. You can do by co-precipitation, ionotropic gelation, microemulsion, reverse micellar model, ultrasonication, microfluidic, and desolvation method. So the chitosan nanoparticle, so you can able to do the drug loading, because this chitosan nanoparticle you can use as a carrier molecule for drug delivery purposes. So these nanoparticles, we can load any type of, we can deliver any type of drug molecule, whether it may be a peptide, whether it may be a compounds. So whether it may be a lipid, whatever it may be, you can you can use the chitosan nanoparticle for carrier molecule. to deliver the drugs into the host cell. So this is while after surface modify the chitosan molecule, so the chitosan molecule is insoluble in nature. Once you surface modify, it is readily soluble in water. It can be able to carry the drug molecule, and you can use it for the drug release mechanism also. So this is the mechanism. The in vitro release, you can so we are specialized in in vitro release as well as in vivo release. In vitro release, there are the criteria in which it depends on release mechanism. One is the pH of medium. If the pH is increased, automatically the drug starts to release. And then polarity of the particle and the presence of enzyme in the dissolution media. So these are the mechanism. So in my study, so how I did this chitosan nanoparticle for various drug delivery application, how these particles were synthesized in different molecules, like surface modified molecules. So I have did the first I procured the chitosan sulfate from the sigma aldrich. So then I there are different mechanisms of synthesis of a chitosan molecule is there. So there was a method which is called Zhu et al. 2006. So according to the et al. Method, I synthesized the N-succinyl chitosan method, N-succinyl chitosan using this method, and CMC carboxymethyl chitosan I used the Wu et al. Method, and quaternary ammonium chitosan and TPP tripolyphosphate chitosan by Hang Liang Zhang et al. Method by 2010. So I have synthesized different varieties of chitosan nanoparticle. There is 4 different types of chitosan nanoparticle. So the defense peptide, so is fluorescein peptide, which I have isolated from the Clarius batrachus non-specific defense system. So that is also I have included after I extracted the peptide which is called fluorescein like peptide from the Clarius batrachus. Then I characterized using the MALDI-TUF matrix assisted laser desorption ionization tandem of light and SDS using this characterization technique. I have characterized the peptide, and the peptide also further purified by the fast protein liquid chromatography, and then I made the peptide lyophilized in the lyophilizer for the peptide source. So this is the stepwise protocol how I isolate. uh, so first crude peptide extraction, then we have to avoid the antimicrobial property, and then using the separate column, I purified. Then the fractions were collected from the fast protein liquid chromatography. Then the different fractions were performed for antimicrobial assay. From that, we identified the specific fraction is having the entire antimicrobial property. So this is the setback purification. This is the stepwise protocol. This is the FPLC which I have done in 5%, 40%, and 80% setback fractions. So then this is the chromatographic chromatogram which I have extracted and identified where the peptide is present in the crude fractions. And I have used two formulas. One is the loading capacity and another is the association efficiency. So whenever if you go for the drug delivery, these two mechanisms are very important. So without knowing the loading capacity and the association efficiency, you cannot able to perform this drug delivery application. This is the figure in which the antimicrobial efficacy was identified in the different forms of chitosan nanoparticle as well as the encapsulated chitosan nanoparticle. So the figure A shows that antimicrobial efficiency of different derived molecule, and the B showed the peptide. Is my slides visible to you? Hello?
No, sir. No, sir.
No, sir, no.
Yes. Sorry for the interruption. Next time I will do from my office where we have the uninterrupted
It's okay,
connection.
sir. No issues,
So,
sir. It's okay.
yeah. Ma'am, now the slides is visible now, ma'am.
No, sir.
Hello. My slides is visible,
Sir,
ma'am.
do one small thing. So you can talk, sir. You have your PPT.
Hello.
Even you can share, sir. You can just speak out from your PPT, sir.
One.
So kindly share your. You can talk, sir.
Yes, ma'am. Yes, ma'am. You, ma'am.
Even if
Now,
slide is not visible, it's okay, sir. You can talk, sir, from your PPT.
okay.
Yeah, now it is visible.
Okay,
Sir.
okay.
Yes, yes, yes,
Yes, ma'am. Yes.
yes.
So this slide shows that the antimicrobial efficacy of the chitosan surface modified chitosan alone, and the peptide which was the chitosan nanoparticle A and B showing when B you found more activity and zone of inhibition when compared to the A. So this is the table shows that the efficiency of the antimicrobial acid zone of inhibition is increased when the concentration of the peptide and the concentration, nanoparticle concentration is increased. The zone of inhibition is also increased. That you can able to observe from the slide. And this is the first characterization in the world. Nobody used the SDS page electrophoresis for characterizing the encapsulation of the nanoparticle, encapsulation of peptide in the nanoparticle for characterizing the both the encapsulation complex with the peptide. So that was the you can able to see in the SDS page, there is a bug in the appearance of the band in the 66 that is the encapsulated peptide with the chitosan carrier, and 25 kDa is the purified peptide. The FR spectrum in which the present slide shows how it was encapsulated, and the peak number are one another, which showed the encapsulation was perfectly done in the C peak. And this slide shows the different time interval, how it was released, and what about the encapsulation efficiency. So this encapsulation efficiency increased.
Sir, not audible, sir.
Is my slides visible to you?
No, sir.
Yes, yes. So that here in this slide you can able to understand the peptide release with respect to the time hours. It shows the sustainable release. See, up
We
to
can see
96
only you.
hours there was a and also in the different concentration also the 1 mg shows the highest maximum efficacy of encapsulation.
The slide
And
is
this
not
slide
visible.
shows the characterization.
Sorry for the interruption, sir. Slide is not visible.
Sorry, extremely sorry. So again and again, this network connections is going bad.
So what should we do, sir? Allow me, sir.
Yes.
Sir.
No, from next time I will do from my office. So even Sunday I will go to my office. Sir. No problem. So this current slide shows that the characterization of the encapsulation by the AFM, that is atomic force field microscope, and the scanning electron microscope. So this slide shows that the encapsulated fluorescent peptide and the free peptide. See, we cannot able to understand whether the peptide was encapsulated properly or not. So in the figure B, you can able to see where the different
Ushasri ma'am.
so only 3 more slides are there, ma'am. Sorry for the interruption.
Yes,
So this slide shows that encapsulated peptide and the free peptide. So in the research, you cannot able to understand whether the peptide is properly encapsulated or not. So this slide shows that it was encapsulated from the B figure, and coming to the conclusion, it is discernible that the synthesized 64 nanometer, the N-succinyl chitosan nanoparticle acts as a good carrier for peptide delivery, and also this NSC, N-succinyl chitosan nanoparticle, along with the peptide which we encapsulated, show a prominent antimicrobial activity, and it is having a good drug response when compared to the standard which was available in the antibiotics such as amoxicillin and the ciprofloxacin antibiotics. So this work was published in Nanobiomedical Engineering, which was the high impact factor journal. So which I published enhanced peptide delivery and sustainable release of fluorescein using N-succinyl chitosan nanoparticle. And a peptide, a patent also made with the peptide, a pharmaceutical formulation exhibiting the antimicrobial activity against the multi-drug resistance bacteria. So I finally thank you one and all for your patience, listening, and if you have any doubt, you clarify with me. And I also especially thank one of our participants who is Professor K. Sambasiva Rao. giant and a star was in the academic and research forum. So, Sir was the previous former Vice Chancellor of Mizoram University and also current Vice Chancellor of Mangal Yadav University. Thank you, sir, for being in this lecture, invited lecture. Thank you, sir. Thank you.
Thank you so much, Bhupesh sir, for your excellent presentation. And I also thank the scientist, vice chancellor, faculty members, students, research scholars for being with us today for this excellent scientific session. And I now request the participants to raise the questions either by raising your hand or putting your questions in chat box. I have few questions, I think, from Prakash sir. What is the size of CNP you obtained, and what is the most desirable size for AMP delivery? So over to you, Bhupesh sir.
Size of the peptide.
Yes.
Hello.
Hello.
The size of the peptide is 25 kDa. 25 kDa. 25 kDa. 25 kilodalton.
Okay, what is the most desirable size for AMP delivery? Can you hear, sir?
Ma'am, actually, yes, ma'am, I'm hearing. Actually, this antimicrobial peptide, so depending on the molecular size, we can go for different size of carrier also, ma'am. So now here the carrier which I use is 64 kDa, 64 kDa size of the carrier that I have used for the peptide which I use for drug delivery. So that it is depending on the size of the peptide which you use, you can use the different type of carrier molecule, nanoparticles.
Okay, sir. There is another question from Dr. Prakash, sir. What is the size of chitin nanoparticle? And yes, sir. Kindly answer this.
Size of nanoparticle?
Chitin nanoparticle.
Yeah. Yes, size of the chitin nanoparticle is 64, sir. 64 kg.
Okay, sir. There's another question from...
that is, here, chitin nanoparticle size is 64 nanometer.
Okay, sir. Thank you, sir. There is another question from Gnana. Signal. What is the temperature sensitivity of this peptide?
Actually, the temperature sensitivity, if you use any compounds, so then there is no need to keep your the formulation in refrigerator centrifuge. Suppose if you are using the peptides like insulin, like fluorosilin or defensive molecule, so then you have to maintain the refrigerated centrifuge like 4 degrees Celsius.
Thank you, sir. Sir, I have one question, sir. You have done lyophilized peptide nanoparticles and also crude. Which one do you find
Yes.
most effective? Like what is the efficacy of these two?
Ma'am, actually, if you go for a pharmaceutical formulation or a drug delivery application, it is always you can prefer the powdered molecule because when you go for a formulation, definitely those molecules we can make it as a colloidal solution. Then only we can able to do those drug delivery application. But so the powdered molecule is used to it is the application of powdered molecule is to transport easily from one another and for until your use, if you use it, if you keep it as a powdered particle, then it is fine. The degradation is very less. But in terms of a solution, if you made as a colloidal solution, then there is a mild auto degradation is there, which I observed from my research experience.
Thank you, sir. There's another question from Rupa. Sir, anti-cancer properties against which type of cell lines? Which cell lines can be used for determining the anti-cancer properties?
Ma'am, anti-cancer property. So we have. So I used the cell lines like this HEBG2 liver cancer cell line, and then
Okay.
HeLa. Those cells I have already experimented. It showed the promising anti-cancer activity.
Okay, sir. Any cytotoxicity that you determined, sir, of using these cell lines?
Ma'am, this cytotoxicity is there, but the cytotoxicity is very meager when compared to that is cytotoxicity in normal cells. It is having very less, but the toxicity is higher in the anti-cancer, that is in the cancer cells. That is the specialty of the peptides, antimicrobials.
Yes, sir. I have one more
But
question. Can
toxicity
I?
is there. The toxicity is there. Yeah.
At what concentration?
Ma'am, up to 30 microgram, I didn't found any toxicity in the cells, ma'am. So above 50, it is showing toxic. It is start to show toxicity in the cells.
Sir, have you
Participants,
studied
any
any?
question?
Sorry,
Yes.
ma'am. Yeah,
Yes,
one
sir.
more from my side. Yes, sir. Have you studied any pathogenic organisms? Have you tried any pathogens against which it is showing successful antimicrobial properties?
Yes, yes, yes. I have challenged against the Klebsiella pneumonia. The Klebsiella pneumonia, which is two type of Klebsiella pneumonia strains I have chosen. One is the urinary tract, which is causing urinary tract UTI, Klebsiella pneumonia, and another one is which is causing the respiratory infection. Both the strains I have tested.
Thank you, sir.
Thank you, Rupa. Thank you, Bhupesh, sir. Any other questions, participants? Prakash, sir. We are much delighted to have all distinguished scientists from CFTRI, Vice Chancellor, sir, research scholars, faculty members, students. I thank you again for being with us on behalf of MBSI. Thank you, Bhupesh, sir, for excellent
Yes,
presentation. And sorry
yeah, yeah.
for the interruptions, anything which happened.
Yes, yes, ma'am.
And, we
Actually,
will
I want
definitely.
to. Yeah, I want to express my sorry to all the participants because
It's okay,
here the
sir. These
network is.
things are common. These
Yeah,
things are
yeah,
common.
yeah.
But in
Yes, yes,
spite of having all these issues,
yeah,
you
yeah.
have
Yes,
carried out and
yes,
presented in a
yes,
well
ma'am.
manner. It was really a good session.
Yes, ma'am.
And thank you all
Yes, ma'am.
for being here.
I Yeah, I especially thank our President, Professor Deshmukh sir, and all the administrative people and also Rupasti ma'am and another one sir who is hosting. So to
Pradhamesh.
all, I especially thank and grateful to you for this uninterrupted, uninterrupted, uninterrupted facilitating this presentation.
You are
Thank
most
you.
welcome, sir.
Yes, thank you.
Shall we wind up, sir?
Yes, ma'am. Yes, ma'am. Yes.
Thanks. Thank you all. Have a wonderful weekend. Thank you.